अस्सलाम वालेकुम। तो आज हम स्टार्ट करते हैं कार्सिनोजेनेसिस को। कार्सिनोजेनेसिस मीन्स दैट हाउ द कैंसर develop, how the normal cell become new plastic are carcinogenic. So carcinogenesis is a multi-step process that involve mutation in the multiple gene. Initially, the new plastic growth is monoclonal in nature, but later on, with time, there are multiple mutations occur. So the new plastic growth shows heterogeneity, and with time, the tumor become more aggressive. And the substances which causes the cancer is known as carcinogens, and they include the chemical substances like cigarette smoking, vinyl chloride, asbestos compound, and the heavy metals like chromium compounds, nickel compounds, etc. And the second one is exposure to the radiation, like exposure to a UV light or X-rays. They causes squamous cell carcinoma or malignant melanoma of the skin. The third one is viruses. So certain viruses are also carcinogenic, like human papilloma virus, Epstein-Barr virus, HIV virus. The next one is the hormonal imbalance, like excessive exposure to the estrogen and decreased activity of progesterone. They lead to the endometrial carcinomas or the breast carcinoma. Similarly, the nutritional factors like high diet rich in the fats and having low fiber content leads to the colonic cancer. And similarly, the cancer can also occur because of the chronic and long-standing inflammation like chronic gastritis leading to the gastric carcinoma. अब बात करते हैं about the hallmarks of the cancer. So the cancer cell shows cell sufficiency in the growth signals. They also show insensitivity to the growth inhibitory signals, and there is altered cellular metabolism occur. Similarly, there is evasion of the apoptotic pathways occur, and the cancer cells are immortal and they have limitless replicative potential. Similarly, they show sustained angiogenesis and cancer cell. There is invasion occur and metastasis occur. Similarly, they have defective DNA repair mechanism, and the cancer cell have the ability to escape the immune responses, and they are not recognized by the immune system. So all of these hallmarks leads toward the cancer. अब बात करते हैं about the genetic defects and the cancer are the gene involved in the cancer. So the first one is the proto-oncogene. Proto-oncogene are the normal genes, and when these genes expresses, they produces proto-oncoproteins, and these proto-oncoproteins causes normal cellular proliferation and the DNA replication. But if there is mutation occurring in these proto-oncogenes, they are converted into the oncogenes, and the oncogenes are carcinogenic in nature, and they causes increase in the proliferation of the cell, and hence they lead towards the new plastic growth. And the conversion of the proto-oncogene to the oncogene, they shows gain of function mutation, and the gain of function mutation shows dominant pattern. Dominant pattern means that they will express if there is mutation occurring in a single allele of a gene, means that if a single allele is mutant, then they will express and they will show overexpression or gain of function mutation. It means that the oncogene will overexpress, and they causes increase in the cellular proliferation. So the mutation in the conversion proto-oncogene to oncogene is gain of function mutation, and it shows dominant pattern. The second one is the tumor suppressor gene. Normally, a tumor suppressor gene controls the DNA replication and the cellular proliferation, and this tumor suppressor gene include P53 gene and the RB genes. But if there is mutation occurring in these tumor suppressor gene, then they will not control the cellular proliferation, and the cell will over-proliferate and will lead towards a new plastic growth. And the mutation occurring in the tumor suppressor gene is the loss of function mutation, and the loss of function mutation shows recessive pattern. Recessive pattern means that if both the alleles of a genes are defective, then the tumor suppressor gene will not express and they will not function properly, and they will not control the cellular proliferation, and there will be poor proliferation of the cell occur, which leads to a new plastic growth. So the new mutation in the tumor suppressor gene is loss of function mutation, and it shows recessive pattern. The third one is regulation of the apoptotic gene. Normally, there is a balance between the proapoptotic genes and the antiapoptotic genes. Like the proapoptotic genes include the Bax and Bach family, and the antiapoptotic genes include the BCL2 genes. The proapoptotic genes, they induces the process of apoptosis, while the antiapoptotic genes, they inhibit the process of the apoptosis. And normally there is a balance between these two genes, but in neoplasia, this balance is lost. The proapoptotic genes, they shows loss of function mutation, so there will be no apoptosis occur. And the anti-apoptotic gene, which inhibit the process of apoptosis, they shows gain of function mutation. So there will be no apoptosis occur, and the cell become immortal and will leads toward a new plastic changes. The fourth one is the defective DNA repair gene. Normally, if there is any damage to a DNA occur, then this DNA damage is recognized by the DNA repair genes, and these genes they correct the mutation or any damage to a DNA. But if there is mutation occurring in the DNA repair genes, and this mutation is loss of function mutation, then there will be no repair of the DNA occur, or the damaged DNA cannot be repaired, and hence the damaged DNA or the mutant DNA will lead to a new plastic growth. अब हम स्टार्ट करते हैं hallmarks of the cancer. So the first one is cell sufficiency in the growth signal. Cell sufficiency means that the new plastic cell they proliferate without any external stimuli or without any growth factors. Normally the cell contain proto-onchogenes, and when these genes expresses, they produces proto-oncoproteins which causes the normal DNA replication and the cellular proliferation. Normally when the proto-oncogene expresses, they produces growth factors, and these growth factors bind with the growth factor receptors and activate it, and the growth factor receptors then activate signal transduction proteins, and these signal transduction proteins include the RAS gene, ABL gene, beta catenin gene, etc. Normally the RAS genes they bind with the GDP and they are present in ectoplasm, but when the growth factors bind with growth factor receptors they are activated, and the inactive RAS gene are converted into the activated RAS gene, and activated RAS gene contain the GTP. Similarly, there is an enzyme known as the GTPase enzyme, and this GTPase enzyme converts the active RAS protein into the inactive RAS protein. When the RAS proteins are activated, which are the signal transduction protein, they activate the nuclear transcription protein like MYC genes, which are the responder genes. And when the nuclear transcription proteins are activated, they activate the cyclin genes. And when the cyclin genes are activated, they form the cyclins. And there are cyclin dependent kinases enzyme in the cell, and these kinases enzymes are cyclin dependent. So when the cyclin proteins are formed, and they combine with the cyclin dependent kinases enzyme, and they form a complex, and this complex then act on the RB gene. Normally the RB gene they hold E2 vector and prevent it from releasing, because the E2 vector causes cellular proliferation, so the RB genes hold it. But when the cyclin bind with the cyclin dependent kinase enzyme and they form a complex, then this complex act on the RB gene, and this kinase enzyme phosphorylate the RB gene. So when the RB gene is phosphorylated, it becomes inactive and it releases the E2 vector, E2F vector. And when the E2F vector is released, this E2 vector causes the DNA replication and the normal cellular proliferation. So in neoplasia, there is either increase in the synthesis of the growth factor occur, or there is overexpression of the growth factor receptors occur, or there is overactivity of the signal transduction protein occur, or there is overexpression of the nuclear transcription proteins or the cyclin genes occur. So the first one is the alter activity of the growth factors. In new plastic growth, there is either increase in the synthesis or the release of the growth factors occur, and these growth factors combine with the growth factor receptors and causes the overproliferation and leads to other new plastic changes. And these growth factors are either released from the stroma, or they are either released by its own from the neoplastic cell. And the neoplastic cell releases growth factors, and these growth factors combine with the same neoplastic cell. So this kind of stimulation is the autocrine stimulation. The example include the mutation in the SIS gene. When this gene overexpressed, they produces platelet-derived growth factor beta, which causes astrocytomas and the osteosarcomas. The second one is the HST1 gene. When this gene overexpressed, are the INT2 genes. When this gene shows amplification, they produces fibroblast growth factors, which causes stomach cancer, breast cancer, and the bladder cancer. And the third one is the tissue growth factor alpha gene. When this gene overexpressed, they produces tissue growth factor alpha, and this tissue growth factor alpha is involved in the hepatocellular carcinomas. The second one is the alter growth factor receptors. Normally, the growth factors, when they are released, they bind with the growth factor receptors and they activate the receptor and causes normal cellular proliferation. But in neoplasia, there is intrinsic mutation occurring in the growth factor receptors, and the growth factor receptor become autonomous in nature, means that they become activated automatically in the absence of any ligand or any growth factors. And similarly, there is either increase in the sensitivity of the receptors to the growth factor. Increased sensitivity means that a smaller number of growth factor can overstimulate the receptors. The third one is the overexpression of the growth factor receptor. So all of these three changes they lead toward the new plastic changes. The first one is the intrinsic mutation in the growth factor receptor. Second one is increased sensitivity, and third one is the overexpression. All of these changes lead to all the new plastic changes. For example, if there is overexpression of the ERBB1 gene, it will overactivate the EGF receptor family, which is epidermal growth factor receptor family, and it will lead to all the squamous cell carcinoma of the lung. Similarly, if there is amplification of the ERBB2 gene or HER2 gene, it will overexpress the EGF receptor family, which are involved in the breast cancer. Similarly, if there is point mutation occurring in the RET gene, then it will overexpress the receptors for neurotropic factors and hence will lead to all the multiple endocrine syndrome type 2 or the medullary thyroid cancer. Similarly, if there is point mutation occurring in the KIT gene, they will activate the receptors for the stem cell. They will overactivate these receptors and hence will lead to all the GI stromal tumors or the soft tissue tumors. To inhibit the overstimulation of the these receptors, we give the target receptor blockers like allotinib or the trastuzumab. So, in neoplasia, there is alteration in the activity of the growth factor receptors occur. There is either autonomous stimulation of growth factor receptor or increase in sensitivity of the receptors occur to growth factors, or there is overexpression of the growth factor receptors, which leads to all the new plastic changes. The third one is the alter signal transduction proteins. And the signal transduction proteins include the VAS gene, which is the most commonly involved in the cancer, ABL gene, beta-catenin genes, and the BRAF gene. So the first one is the VAS gene, which is a signal transducer gene. Normally, the VAS gene is binded with the GDP molecule and is present in the inactiopam. But when a growth factor is combined with the growth factor receptor, they activate the VAS gene. And the VAS gene bind with the GDP molecule and it is converted into the actiopam. But there is an enzyme known as the GTPase enzyme, which converts the activated VAS into the inactiopam and prevents the overactivation of this VAS gene. And when the VAS gene is activated, then it activate a cascade pathway like mitogen activated kinase pathway or the RAP kinase pathway. And these pathway then causes the activation of various nuclear transcription factor like MYC gene, which activate the cyclin gene and causes the normal cellular proliferation. But in neoplasia, there is either the loss of function mutation in the GTPase enzyme occur. When the GTPase enzyme become mutated, then the VAS will remain an ectopump for a longer time and will causes the overstimulation of the nuclear transcription factor and hence causes overproliferation and will lead to other new plastic changes. And similarly, there are the GTPase activated protein, and these proteins bind with the VAS gene and augment the activity of the VAS gene and causes new plastic change. So the VAS gene will overactivate if there is loss of function mutation in the GTPase enzyme occur, or there are the GTPase activated protein binding with the VAS gene and augments its activity. Around about 15 to 20% of the human tumors has the VAS gene mutation, but the frequency of the VAS mutation is much higher in the pancreatic and the colon cancer. Around about 90% of pancreatic cancer has VAS mutation, and about 50% of colon cancer has VAS gene mutation. And the VAS has various variants like KRAS gene mutation found in the colon cancer, the HVAS gene mutation found in the bladder and kidney cancer, and the NRAS gene mutation found in the melanomas. The second one is ABL gene. ABL gene is also a signal transducer gene, and this gene has a non-receptor tyrosine kinase activity. Normally, the ABL gene is present on the longer arm of the chromosome number 9, and this ABL gene from chromosome number 9 is translocated towards the chromosome number 22, where they bind with the BCR gene, and hence there will be ABL BCR fusion occur and they form a chimeric gene. So we say that there is translocation of the ABL gene from chromosome number 9 towards the chromosome number 22, and hence there is formation of ABL BCR hybrid occur, and such kind of chromosome is known as Philadelphia chromosome, and this chromosome has uncontrolled tyrosine kinase activity which gives strong mitotic signals and causes new plastic growth. And the Philadelphia chromosome or the BCR-ABL hybrid formation will leads to all the chronic myeloid leukemia or the acute lymphoblastic leukemia. And such kind of leukemias has uncontrolled tyrosine kinase activity, and such kind of addiction is known as oncogenic addiction. So to treat the leukemias, we give the targeted drug therapy, like we give a drug known as imatinib, and this drug has a targeted tyrosine kinase inhibitor activity, and hence such drug is used in the treatment of chronic myeloid or the acute lymphoblastic leukemia. The third one is the BRAF gene mutation. It is a signal transducer gene, and when this gene expresses, it causes activation of mitogen activated phosphokinase pathway, which causes normal cellular proliferation. But if there is point mutation occur in the BRAF gene, it will causes overactivation of this mitogen activated kinases pathway, and hence will lead toward the overproliferation and the neoplastic growth. And this BRAF gene mutation is found in hairy cell leukemia and the various melanomas. The fourth one is the beta-catenin gene, which is also a signal transducer gene. Normally, when the beta-catenin expresses, it increases the activity of MYC gene, which is a nuclear transcription factor, and this MYC then activates cyclin gene and causes the normal cellular proliferation. To control the activity of beta-catenin gene, we have the APC gene and E-catenin gene. When these gene expresses, they regulate the activity of the beta-catenin gene. But when there is loss of function mutation in the APC gene or the E-catenin gene, then this gene will not work, and hence there will be no inhibition of the beta-catenin occur, and the beta-catenin will overexpress and they causes overproliferation of the cell and will lead towards the neoplastic changes. And the beta-catenin mutation occur in the colon cancer. The fourth one is the nuclear transcription factors. The nuclear transcription factors include MYC gene, MYB gene, JUN gene, and the REL gene. MYC gene is an important and common nuclear transcription factor. The MYC gene, they activate the cyclin gene to pump cyclin, and the cyclin controls the cell growth. And the MYC gene also increases the telomerase activity. And the MYC gene are also involved in the reprogramming of the cell. So the MYC gene are known as master regulator of the cell. If there is mutation in the MYC gene, then they will code for excessive cyclin and there will be abnormal cellular proliferation occur. So the MYC gene, which is present on the longer arm of the chromosome number 8, it is translocated towards the chromosome number 14, where it combines with the IG gene, and the IG gene is active all the time. So there is formation of the MYC IG complex or the MYC IG hybrid occur. So the 8 to 14 translocation is seen in the Burkitt lymphoma, in which there is excessive B cell proliferation occur, and the Burkitt lymphoma is a type of diffuse lymphoma. So the 8 to 14 translocation is seen in diffuse type of lymphoma, and such type of lymphoma is MYC IgG dependent, and 18 to 14 translocation is seen in follicular type of lymphoma, and it is BCL2 dependent. अब बात करते हैं about the subtypes of the MYC gene. So it includes the CMYC, which is involved in the Burkitt lymphoma, NMYC gene, which is involved in the neurofibroma, and the LMYC gene, which is involved in the lung carcinoma. So next is the cyclins. Cyclins are the normal regulators of the cell cycle, and the cyclins are activated by the MYC gene, which is a nuclear transcription factor. And when the cyclin gene they expresses, they produces cyclin proteins. Inside the cell, there are cyclin-dependent kinases enzyme, and these kinases enzyme are cyclin-dependent. When they combine with the cyclin, they form a complex, and these cyclin-dependent kinases enzyme they act on the RB gene. Normally, the RB gene is present in the ectoplasm and it holds the E2 vector and prevents it from releasing. But when the cyclin-dependent kinases enzyme form a complex with the cyclin, these kinases enzyme phosphorylate the RB gene. And when the RB gene is phosphorylated, there is release of E2 vector occur from the RB gene. And when the E2 vector is released, it causes initiate the DNA replication and the cellular proliferation. To control the activity of the cyclin-dependent kinases and cyclin, there are cyclin-dependent kinase inhibitors. And the cyclin-dependent kinase inhibitors has two family. One is the CEPKIP family, and the second one is the human INK4A and the ARF loci family. So the first one is the SIP-KIP family. They include P21, P27, and P57. These cyclin-dependent kinases inhibitors are non-specific in their action, and they can inhibit all the cyclin-dependent kinases enzyme, whereas the human INK4A or the ARF cyclin-dependent kinases inhibitor include P15, P16, P18, and P19. And these inhibitors are highly specific in their action, and they can only inhibit the cyclin-dependent kinase 4 and the 6. There are various types of the various variants of the cyclin, like cyclin D, which is also known as cyclin 4,6, cyclin E, and cyclin A are cyclin 2, and a cyclin B. And these cyclins are involved in controlling the cell cycle at the various phases. Normally, the cell cycle consists of G0 phase in which the cells are present and quiescent are the inactive palm. And palm G0 phase, the cell then enter into the G1 phase in which the cells start preparing for the cell division. Palm G1 phase, the cell then enter into the S phase. S phase is the synthesis phase in which there is replication of the DNA occur. And between the G1 and S phase, there is the first checkpoint. Palm S phase, the cell then enter into a G2 phase, and palm G2, then the cell enters into the mitotic or the M phase. Palm G2 phase to a M2 phase, there is a second checkpoint. So the cyclins are involved in controlling the cell division at various stages of the cell cycle. Like the cyclin D that is involved in controlling the cell cycle or the cell division from G naught phase to the first checkpoint, and the cyclin E which is involved in controlling the cell cycle from the first checkpoint to the S phase, and cyclin A is involved in controlling the cell cycle from S phase to a G2 phase, and the cyclin B control the cell cycle from G2 phase to the mitotic phase. And in between the G1 and the S phase there is first checkpoint, and in between the G2 and the M phase there is the second checkpoint. So the first checkpoint is present between the G1 and the S phase, and if there is any damage to the DNA occur, then this DNA damage is sensed by the P53 gene, and this P53 gene it either activates the DNA repair gene to repair the DNA damage, or if the DNA damage is much severe and there is no repair occur, then P53 activates the BCL2 gene which causes apoptosis and the death of the cell. So the first checkpoint it senses the DNA damage, it either repair the DNA damage by DNA repair genes, or it either, if there is no repair occur, then it causes the apoptosis of the gene, and the DNA damage is sensed by the P53 gene. And the second checkpoint is present between the G2 and the M phase, and the S phase there is replication of the DNA occur. So if there is any mutation in the replicated DNA, then it is arrested at the second checkpoint and between the G2 and the M phase, and it is prevented from entering into the mitotic phase. In neoplasia, both the checkpoints are bypassed by the cell, and the cell enters into the M phase and causes the rapid cell division, and hence will lead to the increase in the cellular proliferation and the new plastic changes. And the cyclin D, which is the first cyclin whose concentration rises in the process of the cell division, and cyclin D is associated with the mantle cell lymphoma. So let's summarize the whole topic. Normally, they are proto-oncogene, and when these gene expresses, they produces the growth factors, which then bind with the growth factor receptors, and they activate the signal transducing gene like RAS gene, and these signal transducing gene then activate the nuclear transcription factors like MYC family, which activate the cyclin gene, and cyclin combined with the cyclin dependent kinases, and they form a complex which then phosphorylate the RB gene, and the RB gene is phosphorylated. It releases the E2 factor, and the E2 factor then causes the DNA replication. But whenever there is mutation in the proto-oncogene, it is converted into the oncogene, and the oncogene then causes increase either in the synthesis of the growth factors, and these growth factors are either released from the stromal cell or from the neoplastic cell by its own. The oncogene they either causes overexpression of the growth factor receptors, or increase in the sensitivity of growth factor receptors to a growth factor, or there is autonomous activation of the growth factor receptors occur in the absence of any growth factor. The third one is the signal transduction gene. So in neoplasia, there is overactivity of the RAS gene occur, there is overactivity of the ABL or the beta catenin gene occur. And similarly, there in neoplasia, there is overactivation of the nuclear transcription factor like MYC family occur, and this MYC family then activate various cyclin, and these cyclins combine with the cyclin dependent kinases enzyme which phosphorylate the RB gene and releases the E2 factor and causes the overproliferation of the cell.